Welcome to Study Loop.
Here for this question, we have three different molecules with different genes being expressed in each of these molecules, or amino acids in the case of the proteins. So our pre-mRNA is going to consist of 5 regions or genes, and this is going to undergo some sort of processing inside of the nucleus to be converted into this mRNA molecule. And this is typically going to include the process of alternative splicing. What this means is part of this gene sequence is going to be spliced or cut out to create this finalized version of the mRNA. And this allows more variability for the types of mRNAs that can be produced from the same pre-mRNA strand. So here in this case, we have spliceosomes, or the proteins responsible for carrying out this splicing. They are cutting out this third gene here, and they are ligating the two fragments together. So you'll end up with this mRNA molecule with genes 1, 2, 4, and 5. From here, this mRNA molecule is going to go to a ribosome, where it's going to be translated into the protein it's going to make, which is going to be including the amino acids from the genes 2 and 4. Likely, you're going to have gene 1 include the start sequence, as well as say some promoters, enhancers, repressors, any kind of modification to the gene sequence, and the fifth part of this gene sequence is going to include that stop codon, telling the sequence to end translation. So here, because the protein consists of primarily 2 and 4, the mutations in either of these two genes are going to be most detrimental to the cell. Because if there's an error inside one of these proteins, this means the protein will function differently, or it could even be non-functional, which can threaten the survival of the cell. So keeping that in mind, the best answer choice here is going to be B, that the mutation that's most likely to affect the cell is going to be a mutation in gene number 2.